I really want to get into the functionality. Okay, so I'd like to start off by just acknowledging a bunch of people who worked on this, specifically Mike and Allison and Renee and Kelly for all his input to the information content of this, and to Nathan Edwards for helping doing a bunch of work to make this GlycoTree sort of seamlessly integrated with GlyGen, and also other people in the GlyGen team who contributed all sorts of other things, which I don't really have time to go into. Just briefly, GlycoTree has a really sort of a long history. Started off in 2003. Got to clear my screen so I can read my slides. With some concepts introduced by Takahashi and Kato. And it's been through various, lots of people have had put input into it, but it hasn't really delivered on its promise until very recently when it became part of the GlyGen project, which is the NIH Common Fund project, which gave it the, sort of the background and infrastructure and funding, everything it needed to do to actually function well. Let's see. Okay, so I'm sure all of you are aware of this, but informatics is challenging partly because Glycan biosynthesis is a really complex process that's not template directed, and so there's lots of things going on in the cell where glycosylation happens, the enzymes that do it, what substrate pools are necessary, all sorts of stuff like that. And so GlycoTree SandBox is really an effort to address this complexity by providing information about the relationships between glycan structure, the enzymes that catalyze glycan biosynthesis, and the reactions catalyzed by these enzymes. And my goal all along has been to make this site something that's accessible to people that don't really have a strong background in computer science, and even that don't have much of a background in glycoscience. And this would be a site where people could learn about these issues or learn about glycans and their functions in biosynthesis. So why do we need the sandbox? Why do we need GlycoTree? Well, part of the reason is that there's a lot of complexity in glycan structure, and since each glycan is made by its own sort of set of enzymes and it's a complex process, we would have to annotate all of the glycans in GlyGen in order to make this information accessible to people. And right now you can see there's almost 16,000 N-linked glycans in GlyGen. And that means that there are about 120,000, at least 120,000 different residues in N-glycans that we would have to annotate. This is just not practical. So we can't do this. So that what we do is we invoke this thing we developed over the years called GlycoTree. And GlycoTree basically uses a parts list to specify structures of glycans. And once it has these parts lists organized in the way we've organized them, this will facilitate biochemical annotation of glycans and the residues in them. So here's a fairly complex, it's complex N-glycan. You can see it's got lots of residues. And basically the way GlycoTree approaches this is to make prototypes or parts that you can assemble to form this glycan. So here we have all the parts of this particular N-glycan labeled with their parts numbers, and basically what GlycoTree does is that it has this big box of parts. You choose the parts you want to put in another box, throw them in the box, and they self-assemble into glycans. And not only do they self-assemble into glycans, they self-assemble the glycans that are semantically annotated with all sorts of information, which is useful. So how does that go about? So here's a close-up on one of the residues in that glycan. This residue is called NC. It is annotated as part of it's an object, a computer object, and it's annotated with these things. It's got an ID, a name, an anomeric configuration, absolute configuration. It's got a ring form, and it's got some assembly instructions. Like this thing, it's called parent ID, says it's connected to this residue NP through site number 4. Now, that's like you go to IKEA, buy a couch or something that says assemble, attach panel A to slot B, right? That's basically assembly instructions for these glycans. And so once you have all the parts, they have embedded in them their assembly instructions. Not only they have embedded in with them assembly instructions, they have embedded with them information about their place in the biological universe. For example, NC is annotated with this piece of information that the enzyme ALG1 transfers this residue to the growing glycan chain during its biosynthesis. Okay, so I'm going to get away from my slides now and actually start showing you how it works. So let's say you're in GlyGen, you're in the GlyGen page, and you've done a search and you've searched for a glycan that you're interested in. It's this one here, this UW glycan. So you can immediately, if that's what you're interested in, is this type of information that's in the sandbox, you click on sandbox, and it brings up this glycan in a particular in this page. Now, the first thing that I would like you to notice is that right up here, and it's not very it's not very much highlighted, but it's really important. This is a thing that tells you what to do. Click on a structure or residue, right? So if you go here, I'm going to explore this glycan. If I go here, I'm going to explore this glycan. If I go here, I'm going to explore this residue, or here, I'm going to explore this residue, right? So if I click on a glycan, you get, so I can click on this one or this one, start on the reference glycan, you get several tables. And I'm going to change that now because I used to think that this related glycans tab was the most important. But especially for new users who don't understand SNFG nomenclature, which this is represented by, I'm going to have it from now on the default place it's going to land is on the residues. And this Residues tab gives you information about all the residues in this glycan. It tells you what they are. It tells you what their residue ID is, and it tells you what they're linked to. Now, up here in the corner here, if you're really interested in this under the hood stuff for GlycoTree, you can click here and you can see now that these residues are labeled with their GlycoTree IDs. So if you're interested in such things, you can go there and think about that. But most people are just going to come here and click and look at these tables. So for example, this one here is called Na, which is this reducing residue here, or potentially reducing. It's a beta-D glucopyranose N-acetyl. And if I'm a naive user, I can see that symbol means that. Or if I'm really don't know, you can see it's got a hand pointer to it, which means it's clickable. If I really don't know much about it at all, I can actually go to PubChem and get some more information about that. So that's the residue table. That is really, I think, helpful for people that don't understand anything about the structures of glycans, what these pictures mean. Also, there is an enzymes tab here, which gives you information about all the enzymes that are directly impact the biosynthesis of this particular glycan, this UW glycan over here. And you can see there's a long list here, and this enzyme list gives you lots of information. Well, it gives you links to the various enzymes and how to find out more about them. For example, if I'm interested in this thing called ALG1, which, as I said before, transfers this residue NC to the growing chain, I can click on its UniProt link and get some more information that way. But as you see, this particular representation just has all these enzymes in a big pile, and it's very useful, but not as useful as if you understood how these residues, how these enzymes related to individual residues. Before I go on, I just like to emphasize here that all this information was compiled by Alison Nairn, and with using information from Kelly and other members of Kelly's lab. And this is, without that information, we wouldn't even be able to start doing this. So this is really the core information that's now being used to annotate these glycans. Okay, so we can see the residues, but even more, if we're looking at the at the site, we can say that it says click on the structure of residue, right? So if we click on this residue here, we get a specific, it becomes highlighted, we get specific information about that residue. And what we know about that residue, especially if you're naive to this, you can see that this triangle, red triangle, is an alpha L-fucose, and it's linked to the residue called Na. And this has been validated by some software we wrote a while back called Curator, and my team did the validation. So we can get information about individual residues here, and the enzymes involved in their biosynthesis, right? So this allows us to drill down into the information about biosynthesis, not just what are the enzymes that are involved in biosynthesis of this structure, but how are they related to the individual components of the structure. And so that was some of the original motivation for forming the site. But the part that I've been working on most recently is this part called related glycans. And so what this does is it takes advantage of the fact that if this glycan is made of these 16 residues, if I can find a glycan that has 15 residues that all are in common with this one here, that would be a potential biochemical precursor to this structure here, because it had just the same thing minus one of the sugars. And in fact, we can look at these biosynthetically related glycans and click on possible precursors. And what we can see in this case is there's two precursors possible. One that lacks the bisecting glucnec, and this one lacks the bisecting glucnec, and this one lacks one of the terminal sialic acids. So let's say that well, I want to make a biosynthetic pathway, and I'm going to end up with here, but I'm going to choose that precursor. Now I can do the same thing with that, and I can look at its possible precursors, and I'll get let's choose another one that's like in another sialic acid. I can go through this and go all the way, and by clicking, I can build up a biosynthetic pathway for this particular glycan. But that's kind of a pain in the butt. It's a lot of work. So what we've done here is sort of automated some of that. So when you get to this site here, if there's enough information, you'll be able to actually show pathways that I've just generated by hand with automatic pathway generation. And here's an example of that. That's this structure we've been looking at. It takes a little time to load, but what you can see here is that the biosynthesis of this glycan can proceed by many different pathways. So each of these rows in this table is a glycan with its accession number, and each of the arcs is a reaction connecting two glycans. So what we can see here is for this particular biosynthetic pathway, which starts at a branch point where complex N glycans biosynthesis begins, so it's a sort of a sub-pathway, and ends in this one that we're interested in. There are 98 structures that could be involved, unique different structures. There are 247 different reactions, and there are 3,900 possible pathways through this maze of biosynthetic interconnections. And you can see there's a little bit of information here saying that there are actually, of things with DP10, there are 20 different structures that this biosynthesis could go through. And so that's kind of interesting. And in fact, we can go through here and we can look at any of these intermediates by mousing over them, get a picture of them. And we can look at any of these reactions by mousing over one of the arcs. And in fact, if we mouse over some arbitrary arc, let's say that one, and we click on it, we can get information about that particular reaction, including the enzymes that catalyze that reaction. Now that's all cool, again, also cool, very nice and useful, but it's hard to navigate. So what we've done is we've introduced this concept called focus. So let's say I arbitrarily go on here and click on that one. I've clicked on that one. Now its focus is that, this RZ glycan, and we can look at specifically the reactions that lead into this glycan, and specifically at reactions that lead out of this glycan to other glycans. And again, we can click on those and get more specific information about it. So that's kind of nice. But now let's say I want to do something else. Well, now I'm kind of stuck on this one here. So I have to drop focus. And now I'm back to the original state where I can look at all of them and without any sort of preconceptions. And so I've got all this information, and it's incredibly complex. But again, it's a little too difficult to navigate through without some help. So what we can do here is go up in the pathways section and choose a particular pathway, unique pathway through this maze of pathways that sort of suits my needs. So the one that's selected now, so there's a selected path now. The selected path is the one that goes through all the intermediates that have checks that are checked here. So this particular pathway goes through these checked these check things, and in fact, I can click on Show Pathway Parameters here, and I can see that these are the reactions in this pathway that go through this those selected intermediates. And in fact, if I want to know the details of that, I can just render the selected path. Now I have that whole pathway described in a fairly concise graphical form that people that don't know anything about computer science can look at and get information on. For example, they can click on the enzymes and get more information about the enzymes. But there are lots of pathways. There are 3,900 pathways through this pathway maze. And so one thing we can do is we can just say, well, I'm not going to select anything. I'm just going to start with a blank slate. And if I click on most likely, that brings us back to the state that we started. That's the default state when you bring up this page. It's the most likely pathway. That is the pathway, the individual pathway that goes through the intermediates that are most visited in the big complex jumble of pathways. But I can say, well, I'd rather do something myself. I've decided, for example, that the very first thing that gets put on this glycan after starting here with the branch point in the biosynthesis pathway, the very first thing that gets put on is a fucosyl residue, which is not too bad, I guess. So I click here, and now you can see it's filled out the whole thing here, all the pathways all the way down, based on forcing it to go through this fucosylated thing. And now when I render those pathways, I can see the first step is fucosylation, and it goes through what it calculates to be the most likely, which is not necessarily really the most likely, but based on the information we have, it is the most likely so far. Another thing we can do here is we can clear the pathways, and we can say, well, I don't want you to help me. I just want to, I want to start here. And if I click here, I can see there are these 5 that I can go to, because those are the 5 that are connected to the first one. So I'll arbitrarily pick this one, select it as in my pathway. Now I have some other highlighted ones. Well, let's arbitrarily pick this one. And then these ones that are highlighted are okay to go through. And I can go as far as I want. And I've selected a bunch of intermediates, and those are now shown here. And if I render this pathway, it looks like this. So there's a partial pathway which I've generated just by clicking and sort of paying attention to what's highlighted. And so now I can say, well, I didn't really mean that. I just rather go back to the most likely pathway. So that's basically the state of the sandbox, the pathway sandbox right now, for enabling people to look at biosynthetic pathways for a particular, in this case, it's limited to N glycans, but obviously, we're going to apply this to O glycans in everywhere we can. Okay, so that's sort of a good overview. But let's think a little bit about some of the other implications of this annotation of these canonical parts in our GlycoTree. Here's a particular glycan, which if you look carefully at it, you can see that it's got something. If you have some background in this, you can see, nah, that's not going to happen. That's a D-fucose. That's really very unusual. It's in all the databases. It's in the GlyGen database, but it's I think people should be aware that aren't really well versed in these things, that that's kind of an anomaly. So one of the things the SandBox does is that when it brings up structures that have such anomalies, the first thing it does right on the top here is it gives you caveats. So since this residue number 201, and I can toggle that, so this is residue 201, is in this glycan, and it has an abiotic configuration. That is, it's D. It should not be D. It should be L. And so that the implication you can infer from that this whole glycan is abiotic. That is, either somebody made this in a lab with chemicals, or they took one from a natural source and made a mistake on characterizing its structure. It's calling that L-fucose D. Okay, so now you can, for example, you can try to show the pathways involved in this, and then it actually does come up with a pathway for this. If I render that pathway, what I can see is that this step that adds the fucose doesn't have any enzymes associated with it, because this is a D-fucose. And so, one of the things we're going to do in the future is to make the annotation here is this, to say not just no enzymes, because that could be because we don't know, but to make this annotation say, this is unusual. This doesn't happen in an organism. This only happens in a test tube. Another type of caveat that we've introduced here is shown in this structure. And what you can see in this structure, if you're familiar with these things, is that there is a glycan linked to the 4 position of this 6 arm mannose. And if you're really familiar with this, you realize that doesn't get it put on unless there's another glycan on there first. The enzyme that put this on has a dependency on structure, which is not in here, right? So if you go to the SandBox and you look at that, you see some caveats here. And here's the one I was just talking about. Residue 31, which is this one here, right? Residue 31 cannot be enzymatically transferred unless residue N9, which isn't here, has been previously added. But it's not here. So this structure is either chemically synthesized or generated from another glycan by hydrolysis of N9 that was there, but it's not there anymore. So now if we look at this, we can try to show pathways, and we don't get any pathways. Now, that doesn't mean there's no possible way to make a pathway, but considering the information we have in GlycoTree, for example, we don't have glycosyl hydrolases, especially this glycosyl hydrolase that would hydrolyze N9 in the GlycoTree database. So you don't get any information about this pathway. But what you do get, right, are these annotations, which, if you're naive, you can learn an awful lot about. And even if you're not naive, I mean, basically, Mike Tiemeyer is doing most of these annotations, and these are things that I learned about by looking at the annotations Mike has made on these canonical parts, glycan parts. And so that's basically it. I don't want to spend too much time, but I'd be really happy to spend any time answering any questions which you might have about this.